Hello, my name is Titia de Lange and I work at the Rockefeller University in New York. And in this lecture, I'm going to discuss how telomeres solve the end protection problem, how they protect the ends of chromosomes. So telomeres are made up of a simple DNA sequence. It's the TTA-GG repeats. So all the telomeres in human cells and in mouse cells have exactly the same sequence, this short repeat, 6-nucleotide repeat, repeated over and over and over again. Thousands of repeats are present at every chromosome end. In human cells, a chromosome end can be as long as 10 kb of telomeric DNA, mouse cells as much as 50 kb. And then at the very end, there is a special structure to the telomere where the G-rich strand, which sticks behind me here, sticks out 3' to the end of the chromosome. And this region is about 50 to 300 nucleotides. And it's very important for telomere function. This DNA is maintained by the enzyme telomerase that needs to maintain DNA because telomeric DNA is lost with cell divisions. So telomerase is a reverse transcriptase that can recognize the end of the chromosome and add, using its template RNA sequences, to the chromosome end. We're not going to talk about telomerase. That's part of the previous lecture, part 1. Today we're going to talk about what these telomeric DNA, these telomeres, are actually doing. They solve the end protection problem, which is a problem that originates from the fact that mammalian cells, like all eukaryotic cells, have a very good system to detect DNA breaks in their genome. So when a DNA break occurs, cells respond to this. They can find the break and activate two powerful transducing kinases, the ATM kinase and the ATR kinase. And these kinases will enforce cell cycle arrest, sometimes even cell death if the damage is very extensive, and they will orchestrate the repair of the DNA break. And of course, the telomere looks just like a broken piece of DNA, and yet these kinases should not become active at the chromosome end, otherwise the cells would never divide. And cells might try to repair their telomeres, which would lead to a mess in the genome. So telomeres must be ignored by the DNA damage response. How does this work? Well, the DNA is inert. It doesn't do too much, but it has this one great ability, and that is to recruit a telomere-specific protein complex that we've called Shelterin. It's a 6-subunit complex, of which 2 subunits bind to the double-stranded part of the telomere, and a third subunit also binds telomeric DNA, but in single-stranded form. We'll go into this in some detail. So this complex was discovered in my lab not as a complex. We just found one telomeric protein, TRF1. We found TRF1 as we were looking for proteins that could bind to telomeric DNA because we suspected that the end protection problem was solved by a protein complex. After we had identified TRF1, purified it, sequenced the protein, and cloned the gene and showed it to be at telomeres, we started looking for other telomeric proteins, and we found one protein that's pretty similar to TRF1, TRF2, that we found in the database and found that it's also at telomeres. And then we found RAP1, this little blue guy here, as an interacting factor of TRF2, TIN2, the yellow protein in the middle, was discovered by Judy Campisi. We then found TPP1 as an interacting factor of TIN2. And finally, POT1, the single-stranded DNA binding protein, was found by Tom Check's lab based on homology to a telomeric protein in unicellular organisms. This is the only protein in Shelterin that is conserved enough to be identified by homology. So let's look at these proteins in a little bit more detail. Let's first look at TRF1 and TRF2, the 2 proteins that bind to double-stranded DNA. They're very similar, and they both bind double-stranded DNA through a C-terminally located MIP domain, a 3-helix bundle that recognizes a telomeric sequence. The MIP domains cannot bind to telomeric DNA by themselves. They can't form a stable complex. They can only bind to telomeres when they're in a dimeric state. And so both TRF1 and TRF2 are dimers, as I tried to draw here, and they're dimerizing through these very large dimerization domains. So these two proteins are very similar in architecture, but they're in fact very different. They interact with different proteins, and they have very different function at telomeres, as you will see. Now let's look at the other DNA binding protein in Shelterin, Pot1, which binds to the single-stranded DNA. Pot1 is also a fairly simple protein. It has a C-terminus with which it binds to TPP1, and TPP1 recruits Pot1 onto the telomere by binding to tin2, which binds to TRF1 and TRF2. And Pot1 has in its N-terminus two DNA binding domains that are very good at binding single-stranded DNA. This structure here from Tom Check's lab shows these two OB folds that are protein folds that can bind nucleic acids and oligosaccharides, binding specifically to the TTA-GG repeats. They can bind this DNA both at a 3' end, as drawn here, but also all along the single-stranded telomeric DNA. So Shelterin is very abundant. It's there at telomeres in all cells. It's ubiquitously expressed. And there are so many copies of Shelterin per telomere that there is enough TRF1 and TRF2 to cover all the telomeric DNA. There's also enough Pot1 to bind all the single-stranded telomeric DNA at the telomere. In fact, there's 10 times more Pot1 than there is room for Pot1 on the single-stranded DNA. But TPP1 and Pot1 are less abundant than the other Shelterin components. And although I don't draw this in my slides, so there are Shelterins that don't have TPP1 and Pot1 on them. Shelterin is highly conserved, and because of this, we can use mice to study Shelterin function, which is a very convenient and good genetic system. The telomeres also contain nucleosomes that I don't draw and Shelterin-associated proteins. So this little protein complex, 6 subunits, has a formidable task. It needs to block 2 different signal transduction pathways that could become inappropriately activated at telomeres, and it has to block DNA repair. So let's first look at the ATM kinase pathway. This is a pathway that recognizes DNA ends. It recognizes DNA ends through the agency of a trimeric complex called MRN, stably binds to the end, recruits the ATM kinase, and the ATM kinase then goes on to phosphorylate proteins in the chromatin nearby, which leads to the accumulation of DNA damaged foci. So the chromatin at a DNA break is altered over many kbs, sometimes hundreds of kbs, and is marked as a site of DNA damage. This will guide repair to that break and control the repair reaction so that the appropriate repair takes place, depending on the cell cycle. I want to point to one protein here, 53BP1, which is a component of these DNA damage foci that we'll use as a marker for ATM and ATR kinase signaling in this talk. So the ATM kinase also, when it becomes active, can enforce cell cycle arrest. It does so through phosphorylation and activation of CHEK2, which then activates P53 and inactivates CDC25 phosphatase, making it impossible for cells to transit from G1 into S phase when DNA replication occurs. So Shelterin blocks this pathway. It also blocks the ATR kinase pathway, a pathway that is initiated when cells have single-stranded DNA at a break. ATR kinase is activated through the agency of RPA. RPA is a very abundant DNA binding protein that binds to single-stranded DNA regardless of the sequence. Once RPA is at one of these breaks where single-stranded DNA is formed. It joins 911, a ring-shaped molecule that loads onto the double-stranded, single-stranded transition, and these proteins then recruit and activate the ATR kinase, which then does pretty much the same thing that the ATM kinase does. It leads to the formation of DNA damage foci that have 53BP1 in them, and it impinges on cell cycle progression through its own effector kinase, check1. So Shelterin needs to repress this pathway. And then Shelterin needs to block DNA repair. And there are various forms of DNA repair it has to take care of. It needs to block non-homologous end joining, a simple ligation reaction that I will go into in some detail. It needs to block homologous recombination at the telomere. And it also needs to prevent excessive resection of the telomere. There is a 5' end here at the telomere where nucleases that like to resect DNA ends could act, and this would, of course, take away much of the telomeric DNA. So how do we figure out how Shelterin does this complicated task, taking care of so many different pathways, a simple 6 subunit complex? Well, we've chosen to study Shelterin in mouse embryo fibroblasts because that allows us to use the genetics of mice to dissect the pathways. Shelterin in general is essential, so you can't make a mouse without Shelterin. It's dead. But you can use conditional deletion in mouse embryo fibroblasts to remove components of the complex. And the way we do this is by using the Cre recombinase. The Cre recombinase can cut specific sites and make a deletion in the genome. So what we do is we take Shelterin encoding subunit encoding genes, we put the Cre recombinase recognition sites in the genes, and then we have cells that contain what we call fluxed alleles that contain these Cre sites in the gene. And we can then take these cells and infect them with Cre recombinase, and the deletion will occur. The gene will be removed. Now, when we do these experiments, we always do them in cells that don't have p53. The reason is that when you remove Shelterin from telomeres, you get a DNA damage response. And in mouse cells, this will lead to arrest or apoptosis. We don't want that because then we can't study what's going on at the telomeres anymore. But in the mouse, if you remove p53, they have no idea that ATM or ATR is active, and they keep dividing. And so this will give us an opportunity to see what the phenotypes are at the telomere. Now, it turns out that nature has designed Shelterin for these kinds of experiments. It couldn't be better. The reason is that the complex is extremely compartmentalized. So as shown here, you can delete TRF2 from the cells, and TRF2 then disappears from the telomeres. But miraculously, the other proteins stay. So TRF1 on its own, it's very similar to TRF2, is enough to recruit TIN2, TPP1, and POT1 to the telomere. The only protein that falls off the telomere together with TRF2 is RAP1. So whenever we see a phenotype with TRF2, we wonder, is this due to the loss of TRF2 or due to the loss of RAP1? And what we do then is we delete RAP1, and if we don't see the phenotype, we know it was a function of TRF2, and it turns out RAP1 doesn't do very much at telomeres. So what happens when you delete TRF2 from cells? Well, there's a really striking phenotype that occurs that you can see in metaphases of chromosomes. So this is a metaphase of normal, healthy mouse cells in which the telomeres have a fully functional Shelterin complex. And what you can see is that these little mouse chromosomes, false colored in red here with a DNA stain, all have little telomeres highlighted by telomeric fish at their end. Now let's see what happens when we delete TRF2. The consequences of TRF2 are quite dramatic. You can see in this slide that most of the telomeres are no longer at the end of a chromosome. They are inside long stretches of DNA. What has happened here is that the cells have mistaken their natural chromosome ends for DNA breaks, got into a panic and started repairing these breaks, and they do it in the only way they can in this situation, that is by fusing one telomere to another. And this is non-homologous enjoining that is taking place. Non-homologous enjoining is a very simple pathway that's very active in mammalian cells, and it repairs DNA breaks. It starts out by the loading of a ring-shaped molecule onto the DNA end, the Q7080 heterodimer. This is a structure of the Q7080 heterodimer on DNA done by Jonathan Goldberg's lab, and you can see that this molecule has an opening that's just big enough to allow DNA to go through. Q7080 can only get on DNA when there is an end. So it normally will not load onto our chromosomal material only when there is a DNA break. It can load, and once it's loaded, it can bring two DNA ends together. This is called synapses. It needs a lot of additional factors to do this, which I won't go into. And then DNA ligase 4 can come in and ligate the two ends together. Ligase 4 also has accessory factors that I won't mention. So this is the pathway that gives rise to the telomere fusions after deletion of TRF2. How do we know this? Well, what we did is we suspected it was non-homologous enjoining. So what we did is we looked at the genetic requirement for Q7080 and ligase 4 in the formation of these fused telomeres. So we deleted TRF2 from cells that either lacked Q7080 or lacked ligase 4, and then the fusions didn't occur. So we know TRF2 itself, not TRF1, TIN2, TPP1, POT1, and also not RAB1, is dedicated to the repression of non-homologous enjoining. But it does a second thing. It blocks the activation of the ATM kinase. How do we know this? Well, when we delete TRF2 from cells, we see that now, all of a sudden, rapidly, the telomeres contain the 53BP1 protein. This is the protein that I pointed out we can use as a marker for ATM signaling. And you can see here that 53BP1 is now at telomeres, co-localizing the green IF with the purple telomeric fish inside nuclei. And this only occurs when TRF2 is deleted with Cre. In the non-Cre situation, when TRF2 is there at the telomere, 53BP1 is not found at telomeres. We know this is the ATM kinase pathway, again from genetic experiments, in which we deleted TRF2 from cells that did not have the ATM kinase. So we use genetics to make such cells, and if we delete TRF2 from an ATM kinase null setting, there is no DNA damage response. So TRF2 then does two things. It's dedicated to the repression of two pathways. On the one hand, the non-homologous enjoining pathway. On the other hand, the ATM kinase pathway. And interestingly, both pathways are initiated by an end-loading factor. Q7080, the ring, in the case of non-homologous enjoining, and the MR11 complex, MRN, in the case of ATM signaling. So how does this work? How does this simple DNA binding protein, TRF2, prevent these end-loading proteins from getting at the telomere? Well, the clue to this came from work that was initially done by Jack Griffith in collaboration with us, who looked at the structure of telomeric DNA in mammalian cells. And he found that the telomeres are not linear, but can attain a T-loop structure. This is an EM, Kleinschmidt spreading, of purified protein-free telomeric DNA from HeLa cells. And you can see that this telomere has a very large lariat structure. Here's another EM analysis, in this case done by Woodcock's lab, in which they analyzed chicken telomeric chromatin. They purified the whole chromatin and could see these T-loop structures. And in fact, telomeres in protozoa and in plants, many organisms are now known to be in a T-loop structure. So what is the T-loop really? Well, we looked into the details of this structure and we surmised that what is really going on here is that the 3' overhang of the telomere strand invades into some point in the double-stranded part of the telomere, forming a displacement loop, single-stranded DNA, because the telomere overhang goes in, so the other strand has to come up, and forming this structure. The point of insertion of the telomeric overhang seems to be variable. Sometimes the loops are big, sometimes the loops are small. But what is constant here is this strand invasion event. And it seems then that telomeres are trying to hide the chromosome end inside the telomeric DNA. Now this links to TRF2 because we also found in collaboration with Jack Griffith early on that TRF2 in vitro by itself can form a T-loop or a T-loop-like structure. So we incubated baculovirus-derived TRF2 with substrates that looked like telomeric DNA and then examined the product by EM, and as you can see here, there are little loops with TRF2 accumulated at the base. More recently, Eric Gilson's lab showed that TRF2 has the interesting ability to wrap telomeric DNA around its very large TRF-H domain. This wrapping of DNA often creates a topological change that may melt nearby DNA, and this could, of course, be very helpful for a protein that is trying to strand invade the 3' overhang. So from these kinds of data, the model evolved that TRF2 represses non-homologous adjoining and ATM signaling by making this T-loop structure, because in the T-loop, the telomere end would not be accessible to the two initiating factors, Q7080 and the MRN complex. So how would one test this? It's not so easy to test, but the idea would be simple. The prediction of the model is that if you delete TRF2 from mouse embryo fibroblast, the loop structure should be lost. So Jack Griffith and I tried to do this experiment for a very long time and failed because the EM technique is not really well suited for quantitative analysis. Fortunately, a new technique was developed by Xiaowei Zhang at Harvard that allowed us to image telomeres based on the telomeric DNA. This is super resolution storm imaging. There's a iBio seminar by Xiaowei on this, and I advise you to watch this. This work we did in collaboration with Xiaowei Zhang, where we used super resolution storm imaging to examine the structure of telomeres in cells with and without TRF2. So Uli Duxani worked with the Zhang lab, examined telomere structure by storm imaging in cells with TRF2, and you can see here some examples of the T-loops he found. Again, there's always a loop and a tail, but the ratio of the DNA in the loop or the tail can be very variable, allowing us to conclude that it's not so much the loop that's important for the protective activity, but it's the strand invasion event. So then Uli wanted to image cells that don't have TRF2, but here there is a problem, because if you delete TRF2, of course, all the telomeres fuse together. And if you don't see T-loops at fused telomeres, it's not a very informative experiment. So we used a genetic trick to delete TRF2 from ATM null cells in which case there is no DNA damage signaling at the telomeres, as I showed you, and also no fusion. And this is what Uli saw. The telomeres in these cells that lacked TRF2 but had all the other sheltering components there had a linear structure. The T-loop was lost. He then repeated the experiments in TRF1 knockout cells, Pot1, etc., knockout cells, and the T-loops always remained. Only when TRF2 is removed do the T-loops resolve. And so this is fitting with the model. The idea is simply that without TRF2, the telomere can no longer maintain the T-loop state. Now becomes a linear molecule that allows Q7080 to load onto the end, and you get non-homologous interjoining. Furthermore, the MRN complex can now see this telomere for what it really is, a DNA end, load onto the end, and activate the ATM kinase, leading to the 53BP1 foci at the telomere. So this takes care of one major problem in telomeres, the ATM signaling pathway and non-homologous interjoining. What about the ATR kinase? Again, this is a very powerful kinase that could kill the cells if it was activated at the telomeres. It is repressed by another sheltering component. It's repressed by Pot1. So Pot1, the single-stranded DNA binding component in Shelterin, is the one that represses the ATR signaling pathway. How do we know this? Again, we used the genetics in the mouse and looked at what is going on at telomeres when Pot1 is there. This is cells with an intact sheltering complex, and there are no 53BP1 foci at telomeres. There's no DNA damage response. Now we delete Pot1, and immediately you see that there is a lot of BP1 at co-localizing with the telomeric DNA. This is the ATR kinase pathway that has become active at these chromosome ends. How do we know that? Well, we used the genetics in the mouse again and now deleted Pot1 from cells that do not have ATR. And if you do that without the ATR kinase, no 53BP1 foci at the telomere. So this told us that while TRF2 is dedicated to the ATM pathway, Pot1 is dedicated to the ATR pathway. I should say, parenthetically, that all these experiments were done in mouse cells where there are actually two Pot1 proteins, but for simplicity, I refer to them as Pot1. How does Pot1 repress the ATR signaling pathway? Well, we got a clue about the mechanism from looking at the RPA protein that I told you is important to activate the ATR kinase. So when you delete Pot1 from telomeres, you all of a sudden see RPA accumulate at the telomeres, right there. Normally, no RPA at telomeres. So when Pot1 is removed, RPA can bind to the single-stranded telomeric DNA. And this made us think that what really is going on here is that Pot1 bound to the telomeric DNA is normally excluding RPA from the telomere, and since RPA can't bind, it can't activate ATR. So the idea is that the whole single-stranded part of the telomere here is covered by Pot1, and then RPA can't bind. But there is a problem with this model, and that is that RPA is extremely abundant. So this is a competition model where Pot1 wants to win in this competition from RPA, but it's dealing with a protein that's about 200-fold more abundant than itself. Not a good situation. Furthermore, we found that RPA and Pot1 have exactly the same affinity for the telomeric DNA. So Pot1 doesn't have an advantage at that level either. So how then can this simple single-stranded DNA binding protein prevent RPA from binding? Well, the trick here is that Pot1 is tethered. So Pot1, after all, is tethered onto the telomere by TRF1 and TRF2, so that if one of the pot1s on the single-stranded DNA lets go of the DNA, first of all, the DNA is held there by other pot1 proteins, and secondly, the pot1 that has released is still sitting there because it's tethered to TRF1 and TRF2 through TPP1 and tin2. So it can rebind immediately. Whereas RPA, if it lets go of the DNA, like any DNA binding protein will diffuse away and it has to start all over. So the prediction of this model is that the tethering of pot1 to the rest of Shelterin is critical for the ability of pot1 to protect the telomere from ATR signaling. And that's a testable prediction. So what we did is we created situations where not just pot1 is deleted, in which case the ATR kinase becomes active, but we leave pot1 in the cell and ruin its tethering. We can do that by deleting TPP1. We found that ATR becomes active. Or we can do it by deleting tin2. We found that ATR becomes active. And probably the best experiment of this set was an experiment in which we took the tin2 knockout cells and put back into them a mutant form of tin2 that's completely normal, can do everything except for binding to TPP1. So these cells now have all Shelterin components there, and yet, because pot1 is not tethered to the telomere, ATR becomes active. So the tethering of pot1 and the repression of ATR signaling is critical for telomeres. Otherwise, this signaling pathway becomes active. And it's not only important for telomeres that are in the open configuration as drawn here. Telomeres may be open when the T-loop opens up. They may be in linear structure when the T-loop opens up during DNA replication. But the same repression of ATR signaling is important when telomeres are in the T-loop configuration. Because in the T-loop configuration, at the very base of the T-loop, you have a structure that is a perfectly fine substrate for ATR signaling. It's a structure that has single-stranded DNA in the D-loop where RPA can load, and it has a 5' end here and a single-stranded, double-stranded junction, which allows 911 to load. So everything here is set up to activate ATR, and also, therefore, in this setting, pot1 needs to be able to repress the ATR signaling. T-loops themselves don't do anything to repress ATR signaling. When we delete pot1, the T-loops are still there, but the absence of this single-stranded DNA binding protein at the base of the T-loop creates the trouble. So, in summary then, I discussed with you how telomeres solve the basic tenets of the end protection problem. Telomeres are made up of a long stretch with very boring repeat sequences. This is important because it provides many binding sites for a Shelterin, the 6 subunit complex that's recruited onto the telomeric repeats. And Shelterin then has 2 main factors that do the job. On the one hand, there is TRF2. It creates the T-loop structure, and this represses non-homologous end-joining and ATM signaling. On the other hand, there is pot1, which by its ability to associate with the single-stranded DNA, can repress ATR signaling by keeping RPA off the telomere. Now, I've discussed this in some detail, but I must say there is much else about telomeres that I haven't been able to discuss, and I can't leave you with the idea that this is everything. So I'm just quickly going to list the many interesting subjects at telomeres that I haven't had time to go into. I haven't talked to you about TRF1. It's the first telomeric protein we found. It turns out not to be involved in the end protection problem. Instead, it's a protein that helps the DNA replication fork go through the telomeric DNA, which is extremely difficult for the replication fork to negotiate. I haven't told you how Shelterin creates the 3' overhang. That's so important to make the T-loop structure. I haven't told you how Shelterin represses excessive 5' end resection, which threaten telomeres, or about homologous recombination, or about the real danger of T-loops, which have a structure at their base, at the tail loops junction, that can be resolved by holiday junction resolvases that could cleave off the whole loop if they act. I haven't told you about alternative non-homologous end joining and PARP activation at telomeres. And lastly, the whole story I told you about telomeres is a story of DNA and protein, but it leaves out one component. It turns out telomeres are transcribed into a long non-coding RNA, link RNA, called TERA, that undoubtedly has an important function at telomeres. We just don't know yet how it works. So I want to say that I have discussed work from my lab, and I haven't had the opportunity to mention everybody that contributed to this work. And so if you're interested, people from our lab, please go to our website, the Lange Lab at rockefeller.edu, where you can find all that information, both the current members of the lab and the alumni. And it's a fantastic group of people that I've been fortunate to work with over the years. It also lists our collaborators. It lists our protocols, if you're interested in doing these kinds of experiments. It lists information on how to get our reagents in the lab, where to get our constructs are on Adgene, our mice are in Jackson Lab. The MEF cell lines will be available at the ATCC, so you have a lot to find in our website. We do our work at the Rockefeller University, which is a great place to work in the Upper East Side of Manhattan. It's a beautiful campus and a wonderful collegial environment, and we're currently supported by the funding agencies mentioned here. So thanks very much for listening, and it was a pleasure to do this.